There we are. It is October 10th. This is Biology 105. We're going to wrap up our week 6 PowerPoint today, make some progress into our week 7 PowerPoint, and before you know it, we'll be right on track with everything. Okay? Don't forget, folks, next week you've got that online lab midterm. Okay? Online lab midterm right here. 50 points. Why is it not? Oh, yeah, because I made it in new quizzes, so it's really annoying. It won't just show the preview. I have to hit the build button. So it covers weeks 1 through 7 in your lab. So do it after, week 7. That's this week, so you'll be fine there. It's not opening until after then. Anyways, yeah, you have 1 attempt. You have 2 hours to complete it. It's all multiple choice and drop-down windows, but the actual lab final will not be all multiple choice and drop-down windows. It will have, prompts with short answers being requested. Basically, like at the end of all your protocols. Questions about the upcoming lab midterm? Oh, you can even see the first question there. How many milliliters are in 4.5 liters? Don't answer that, but yeah, question. One second. No, it's totally fine. 11. Yeah. It's just meant to get you, to check in and make, yeah, you got it. Of course. Boom, boom. Any other questions about anything in the course? Otherwise, we'll just get started with lecture stuff. Okay, cool. The only thing I wanted to talk about with enzymes that we didn't get a chance to talk about yet is cofactors, okay? They'll come up in your lab. Cofactors, they are not proteins, but they are required by an enzyme to function. So remember, enzymes are proteins, all right? But sometimes they incorporate something else that's not a protein that helps them function. These are called cofactors. Specifically, if the cofactor is an organic molecule, so it's carbon-based, like all these molecules on the right here. Oh my gosh, 1 second. Like all these molecules here, okay? These are all organic molecules, large carbon-based, all right? We call organic cofactors coenzymes. They're still cofactors, okay? And many vitamins that you consume are examples of coenzymes, all right? And the term, yes, please. Oh, they could be made out of ions, right? That's the most standard issue cofactor. It's like a magnesium ion or an iron ion. Those are really standard issue ones. Yeah, no problem. Good question. Very good. Okay, cool. So what's a vitamin? A vitamin is something that you supplement, you need it, but you don't make it or you don't make enough of it for normal function. All right, that's the definition of a vitamin. So it's not that coenzymes are vitamins, it's that a lot of the vitamins that you take are coenzymes, and that's their function when you take them. Cool? Sometimes people will say, oh, but vitamin D, like why is vitamin D on there? We make vitamin D when we're exposed to sunlight. But it's sort of a misnomer. It's not technically a vitamin because you are capable of making it, but you don't make enough, you supplement it, and calling things a vitamin is a great way of, marketing them. Anyway, that's all the enzyme talk. We're going to get into respiration. Question? Totally. So, yeah. No, it's fine. It's fine. Yeah. Let's open this up. It's one of these slides here somewhere. What determines whether enzyme inhibition is reversible or irreversible? So essentially, if you have an inhibitor that permanently binds the enzyme and can't fall out of either the allosteric site or the active site, then the enzyme's inhibited, irreversibly. Also, if you use a chemical to inhibit an enzyme that denatures the enzyme, that's irreversible. That's irreversible inhibition, sort of. Yeah, not substrate. Inhibitor. Yep, which is, yeah. It could be a competitive inhibitor, it could be a non-competitive inhibitor, but if it binds to the enzyme and then it stays there, then that thing is going to stay inhibited.
Okay, but unless it has that, they'll all be reversible.
Yep. And usually the inhibitor binds and then naturally just falls off, and binds and then falls off. That's more the natural system. But we can generate mimics of those inhibitors that are designed to stay bound if you want it to be a more irreversible inhibition, like with something like glyphosate. Yeah, no problem. Okay, sick. So now we're going to talk about cellular respiration. And cellular respiration's really, it's an enzyme question, all right? So what we've got when we're looking at cellular respiration is we're going to be focusing on cellular respiration in eukaryotic organisms. And in I'm going to close this door. I don't know what is going on out there. Somebody's tearing apart a chair or something. Trying to get a free soda out of a vending machine, I don't know. But whatever it is, it's a ridiculous amount of noise, and I won't stand for it. All right. Respiration, ready? In respiration, you take high-energy organic molecules and you break them down and you generate some ATP, and then you get that ATP and you use it to do work in the cell, right? Fair enough. So we're talking about cellular respiration in eukaryotic organisms, and don't worry, we'll come back to this. And in eukaryotic organisms, cellular respiration all goes on in the mitochondria, okay? At least the key steps. It's all about compartmentalization of enzyme activity. So you have all these metabolic pathways responsible for cellular respiration, and you put all of the enzymes involved in cellular respiration into a mitochondrion, and that means that you're doing it in the mitochondrion. Fair enough? Okay, great. Now there's going to be some clunky stuff here. That's why we've got a whole PowerPoint just for cellular respiration. So bear with me. Bear with me, and we'll get through it. ATP, all right? Cellular respiration exists to generate ATP. There are also breakdown products used in other biochemical pathways, but cellular respiration is used to generate ATP. All right. Now, there are two ways that ATP gets generated during cellular respiration. One is called substrate level phosphorylation, where there's an enzyme and it binds a substrate, and this substrate that it bound has a phosphate group on it. All right? So it's substrate level. It takes that phosphate group, pops it onto ADP, and that makes ATP. So this is a direct transfer of phosphate from one molecule to another. It's called substrate level phosphorylation because there's an enzyme that takes a phosphate from a substrate and attaches it to ADP, which is actually a substrate as well for it. But this isn't the main way that you get ATP, or it's not where you get the majority of ATP from cellular respiration. Instead, the majority of ATP is generated via this process of chemiosmosis or oxidative phosphorylation. Okay? Oxidative phosphorylation is going to be our key term here. Don't worry, this is all going to come back in detail. I'm just trying to lay the groundwork. So check it out. Here's a substrate level phosphorylation example. you've got this molecule. It's pretty high energy. It's got a phosphate group. Here's the enzyme. It binds PEP. It binds adenosine diphosphate, and it transfers the phosphate group directly. You don't have to know these examples or anything, all right? It's just that we talk about substrate level phosphorylation, but we usually don't go into any depth on it, and I wanted to add in a couple of slides where you saw an image of substrate level phosphorylation. So maybe it'll make more sense as we move forward. Okay. We are not going to do this. I've decided right here. We're going to talk about glycolysis. And basically, when we talk about glycolysis in Bio 105, we just talk about glucose. Glucose gets split into pyruvate. And you get a little bit of ATP from that split due to substrate level phosphorylation. But here's what's actually going on in that process. There's this wild metabolic pathway that goes on where you phosphorylate some molecules, and then you convert them, and then you yank those phosphate groups back off of these molecules. It's a whole nightmare situation that's very complicated. And this here is a simplified glycolysis pathway anyways. So the key here is that you're going to, in this process called glycolysis, you're going to take a big complicated molecule, break it down into simpler molecules, and you're going to get a little ATP out of it. All right, question?
Does it only work if it starts with glucose, or could you like start it with fructose?
Great question. Yes, you can funnel different molecules into these pathways, right? You can push something in later into the glycolysis pathway. Absolutely. You just usually get a little less energy out of it. But this is just the first step of respiration, and you've barely harvested any of the energy initially available in glucose. So if you start with fructose, you're still going to get a decent energy yield out of breaking down that molecule. Solid.
Do you have a picture of the more complicated, unsimplified glycolysis?
No, I do not. But I encourage you to look at them. They're terrible. They're terrible, they're intimidating. This is a pretty good one. It's not, I wouldn't say it's oversimplified, it's just simplified. This is a pretty complete glycolysis pathway. Yeah. Good. Okay. The other way that you get ATP is this process of chemiosmosis. Now remember, phospholipid bilayers are impermeable to protons, all right? They don't cross. But we've got these proton pumps, remember those, the transport proteins, that serve to pump protons in one direction. So check this out. One way to store energy is to pump a bunch of protons in one direction and set up a concentration gradient, all right? This concentration gradient is a form of energy. It generates voltage, right? And then the special enzyme here called ATP synthase, which it has the ACE suffix, so it's an enzyme, and it synthesizes ATP. It's ATP synthase. It's a very friendly name for that enzyme. What this does is it acts like a dam, all right, where protons can flow through it, and that makes it spin. And as it spins, it changes shape. And as it changes shape, it changes activity. And what it does is it takes ADP and an organic phosphate, and it makes ATP. The energy source is actually the concentration gradient, okay, across that membrane. We are going to talk more about what pumps the protons, but I just want you to see substrate level phosphor... Oh, I don't have my clicker working. I want you to see substrate level phosphorylation and chemiosmosis. So we got substrate level phosphorylation and then chemiosmosis. And this is where the majority of the ATP generation is going to come from. And this is why I was saying in the membrane PowerPoint, hey, if membranes, phospholipid bilayers, become permeable to protons, then you die, all right? Because you can't generate any of this ATP. And without this source of ATP, we can't survive. We cannot survive on substrate level phosphorylation alone. It's just a shame, I know. Great. Structures, order, compartmentalization of enzyme activity. Good? So yeah, awesome. This person is running and they are respiring. They are breathing in oxygen, they are breathing out carbon dioxide. They're also carrying out cellular respiration in their muscle tissue. They need ATP to fire off those muscles, all right? Getting that ATP by breaking down glucose and other organic molecules, all right? Reacting them with oxygen essentially, and then producing carbon dioxide, water, and ATP. This is an exergonic reaction. This is a catabolic reaction, all right? This reaction here goes from high energy reactants to low energy products. Therefore, it has a negative delta G value. It's trying to refresh all of our energetics talk that we've been talking about. All right. Hey, you know how fire gets extinguished if you'd starve it of oxygen? Well, cellular respiration is like slow fire. All right. If there's no oxygen available, the process can't go forward. That's why you need oxygen. Okay. How efficient is cellular respiration? It's okay. It's pretty good. 35 to 40% of the energy in the glucose molecule can be harvested. That's not too bad, all right? It's better than a dumb gasoline engine, right? But gasoline engine makes a lot of cool noises. It's pretty cool, you know? We just like talk and stuff, so I'll take the engine anyway. Check it out. This is an engine example. It's always used. I heard it back when I was in school. You probably heard it in other classes. Maybe you heard it in high school. If the gasoline engine was 100% efficient, it would not get warm. If it was 100% efficient at converting the energy, the potential energy inside of the fuel molecules into kinetic energy of movement, which is what it's designed to do, then it would not heat up. The inefficiency is usually just represented by heat radiating out of the system. And the same thing is true with cellular respiration. So 35 to 40% efficiency and taking that energy and moving it from glucose to ATP means you're going to release about 60% of that energy as heat. Now, usually that's considered as a byproduct of cellular respiration, but sometimes you intentionally carry out cellular respiration in order to get the heat byproduct. Like when you shiver, all right? When you shiver, you're firing your muscle tissues so that you're burning a lot of glucose so that you generate a bunch of heat. Cool? Question?
Even though 60% of the energy is lost in the form of heat, can your body or organisms, in this case, use that heat to carry out other chemical reactions, mainly endergonic reactions that require heat?
So not usually. Beyond when a system has more energy, like ambient energy in it, the reactions are more likely to happen because it's easier to get to those, say, transition states of exergonic reactions. But you're not usually able to drive those endergonic reactions with the heat lost here. So that's included in delta G values, where the delta, this is getting a little bit weird with it, but the delta G value going from, my clicker doesn't work, I forgot. The delta G value of, good golly, this reaction, all right, should only include the energy release that's available to do work and excludes the energy radiated out of the system as heat. All right. So the answer is no, you usually can't use that energy for anything, but having more movement of molecules going on in the organism makes reactions happen faster. So depending on how you define it, but it wouldn't contribute to reaching the delta G value for an endergonic reaction. Does that kind of make sense?
Yeah.
Okay, cool.
I'd actually say yeah, it does. So the heat isn't really that useful unless in specific scenarios.
Yeah, I mean, you got to stay warm to live. So that's useful, like maintaining your temperature. But you know, you're not, it's energy that then you're just making so everything keeps moving in your body. Yeah. Yes.
Do you know if the body has any other forms of heat generation? Or if it's just ATP?
Oh, I mean, like your body can just ramp up metabolic rates when you are colder without necessarily just like firing muscles. But shivering is a way to get the muscle tissue to fire more rapidly, and it usually ramps up cellular respiration. It's super rapidly. So there are other mechanisms where if you're just cold, your metabolism goes up. It's a way that your body tries to maintain homeostasis. Yeah. Now, as far as the details of those processes, I'd love to know more about them than I know. But that's the truth about most of reality. I'd love to know more. All right. So cellular respiration. Cellular respiration takes energy from glucose. It's potential energy, it's chemical energy, and it banks it in these ATP molecules. Why don't we just break down all the glucose and just keep a ton of ATP in our cells? Well, because ATP is not as stable as glucose. It's certainly not as stable as glycogen or starch, all right? And you don't really want a ton, you only want these unstable ATP molecules when you need them, right? Otherwise, they can be wasted. I'll never test you on the number of ATP molecules produced by cellular respiration, and I won't test you on percentages like this, about like what percentage of the energy in glucose is actually converted into ATP. I don't think memorizing numbers like that is a good use of your time. You can always look those things up if you become a biochemist, you know? And also, this is an average. It's nowhere near accurate, right? It doesn't represent the variability at all. Anyways, the number 32 ATP molecules, it'll be relevant because you're going to see like, oh, you get 2 ATP molecules here and 2 ATP molecules here and 28 ATP molecules here. But the important thing is where the majority comes from and where the minority comes from. Where do you get most of the ATP molecules from? Okay, and that's going to be chemiosmosis. Solid. Or oxidative phosphorylation, more specifically, because oxidative phosphorylation is the whole net process. But don't worry, I've got that coming towards you on slides. Yes?
I don't know if you're going to cover this, is what you just said, but regarding the word you just said, oh my god, does it always have to be hydrogen that is contributing to the gradient?
My understanding, I know I totally understand what you're saying. Yeah, no, I totally hear you. For ATP synthase to operate, it works with hydrogen, right? I would bet you dollars to donuts that there's something that can use a similar enzyme, all right, with a different ion. I would just assume that's the case, like potassium or something. With ATP synthase, this is going to be with hydrogen.
Okay.
Yeah.
Make sure there weren't any.
No, totally.
Okay.
Absolutely. Oh, yeah, no problem. No problem at all. All right, so we've talked about energetics of reactions. We've talked about endergonic, exergonic, anabolic, catabolic, right? Positive delta Gs, negative delta Gs. These are all ways for us to discuss energetics, okay? Now we're going to talk about energy transfers that specifically have to do with electron movements, okay? These are called redox reactions because redox is a portmanteau of reduction and oxidation. So I'm going to use my thermos. Since I'm holding my thermos, I'm going to enjoy a brief respite and drink some coffee. Splendid, as always. All right. Now I'm holding it. Now it's an electron, so I can't drink it anymore. All right? Look at this electron. I've got it right here. If I've got an extra electron, that means I could have another, say, hydrogen atom attached to me. And that means that I'm a bigger molecule with more order and more energy. Fair enough? So if I take that electron and I put it onto something else, I've just given it some energy. Because now it could form a new bond, maybe with a hydrogen, maybe it just has a new covalent bond form. So I'm transferring electrons and transferring energy. Reasonable enough? If you take the electron on, you have a little more structure, a little more order. You could bond to a hydrogen or something else. So that means that you get more energy in your structure. So far, so good? So here I am, I'm higher energy, I've got this electron. Now I'm gonna go over and put the electron on that table. Here I go. It's gonna happen. It's about to happen. And now, redox reaction. And a redox reaction. And a redox reaction. The electron transfer is a redox reaction where one thing gets reduced, it receives the electron, and another thing gets oxidized, it loses the electron. Bless you. No problem. So, oxidation is loss of an electron, reduction is gain of an electron. If you need to remember, if you need a mnemonic device, remember that electrons have a negative charge. If you gain an electron, you get reduced. Cool? Awesome. Now, sometimes redox reactions are taught as transfers of hydrogen atoms or hydrogen ions, right? Because the electron usually brings a hydrogen with it. Or once the electron's attached, a hydrogen usually just attaches to it, because there's lots of hydrogen ions in the solution. Because of the dynamic nature of water and its slight reactivity and how pH exists, and there's always some hydrogen ions floating around. Solid. All right, cool. So, I'm going to do it again. Here I am. I transfer my electron. The redox reaction occurs. The table became reduced, and I became oxidized. Awesome? I mean, awesome. Yeah, I guess. You know. So one last piece of this, one last piece of terminology. When I am reducing something else, I am termed the reducing agent. So I come over and I reduce the table by attaching an electron to it. I was the reducing agent. But in that reaction, the table oxidized me. It was the oxidizing agent. Okay? Does that sort of make some sense, folks? Was that a no? I thought I just heard somebody say no. which is always okay to say. That's why I ask. Let's just do a quick summary of it. All right. Here's an electron. There's an arrow showing the electron transfer. It's my finest work. Redox reaction. All right. Here's a molecule here. We'll just call it molecule A and molecule B. An electron goes from molecule A to molecule B. All right. A was oxidized. All right. B was reduced. Okay. A equals the reducing agent. Bless you. B equals the oxidizing agent. Okay? Oh, let me put it on the main screen because people might be watching the recording. And there's a summary of what I've been talking about so far. So it's called a redox. There's always a reduction and an oxidation. Are there any questions about this? Awesome. Let's keep on keeping on. Here's cellular respiration as a redox reaction. Okay? Check it out. Glucose is going to get a bunch of electrons stripped off of it. It's going to be oxidized. Those electrons are going to end up on oxygen. It's going to get reduced. As oxygen gets reduced, it takes on hydrogen ions, and it becomes water. As glucose gets oxidized, all right, and electrons get stripped off, this breaks covalent bonds in glucose's structure, and it breaks down to carbon dioxide. Cool? All right. Oh, I just wish I left the receiver. Yes, a question.
So can you explain which one is the oxidized and which one is the reducing agent?
Yeah, totally. So those actually, they're not really fully illustrated on this slide because there's going to be these key players of like NAD plus and NADH, which are going to be the intermediate electron carriers and are actually going to carry out the redox reactions. So no, to answer your question. Well, I'm capable of it, but I'm going to choose not to because, as you can already tell, it will overcomplicate things. I'll do it when we have a diagram that shows them.
So regarding cellular respiration, don't worry about reducing oxidizing agents?
Worry about what is becoming reduced and what is being oxidized, but don't worry about the reducing agent and the oxidizing agent right now, because these big arrows indicate a ton of steps with different reducing and oxidizing agents. Is that satisfactory?
Yes.
Okay, I'll take it. Awesome. Okay. In fact, this slide kind of brings that up. It's not a straightforward, like all of the electrons go slam onto oxygen all at once. That's what happens in an explosion, okay? Where all of the electrons get ripped off of something all at once and then fall to a super stable state. If you watched the movie, let's be honest, the movie The Martian, right? You see that movie?
Yeah.
Thank you. Very good. He eats the potatoes. He eats the poo potatoes. It's great. He doesn't have he makes ketchup. He runs out of ketchup. It's a terrible thing. He listens to music. He drives around. It's the whole thing. All right. So he blows up a bubble because he's trying to make water. He's trying to carry out this reaction, reacting hydrogen gas and oxygen, and just getting those to fall to this super stable state, and boom, explosive release of energy. Okay? Now, essentially what we're going to be doing in cellular respiration is this reaction, but in a very, very controlled way. Because you don't want one molecule of water to get produced from these, because it will explode and provide the initial activation energy for continued reactions here. All right? Because this is an exergonic reaction. It's energetically favored, but it's not spontaneous. But if you constantly provide activation energy, it just keeps exploding. Okay? Cool? That's why when you light a log on fire and it finally starts to burn, it hits like this threshold where it's providing enough activation energy for everything else to burn, and you start a chain reaction, and it can be bad news. So in cellular respiration, we've got a very controlled reaction where these electrons are going to start at a super high energy state, and they're just going to fall boop, 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 down to a low energy state, but step by step by step by step, because if the electrons went from high to low all at once, you'd explode. Okay? Cool? Don't do that. Not in my class. All right? Thank you. I'm constantly focusing on it every day. It's working, apparently, because I haven't done so yet. So we're going to talk about this electron transport chain, which is exactly what I was talking to you about a moment ago, right? How we're going to be taking these electrons and doing a ton of redox reactions with them. And every time we do another redox reaction, we release a little bit of energy, and the energy released by this series of redox reactions is going to be used to pump protons, actually, which then will flow through ATP synthase to generate ATP. I've got some better diagrams coming up with more detail. I'm just introducing the concept right here of the multi-step pathway, like what we talked about in the dang enzyme thing. It doesn't matter what I said. Go back and watch the recording for the Easter egg. Got to play it backwards. What I said was, it's like what I said in the enzyme. I said something about the enzyme PowerPoint. I don't even remember what it was. It's Friday. We only got about 12 minutes left in this lecture. Yeah, but don't worry. You're gonna get the best, the best performance out of me that you deserve. Here we go. Here's a mitochondrion in the diagram, and then an actual image. Cool? Remember mitochondrial structure? We talked about it. We talked about endosymbiosis. We talked about a bunch of enzymes, right? I'm sorry, a bunch of membranes. Each of these membranes is a phospholipid bilayer, so it's impermeable to protons. That's the key thing to remember right now. Because this is all about the cellular respiration. This whole pathway is all about pump a bunch of protons and then carry out chemiosmosis to get ATP. All right? We pump protons across these membranes, and hey, if you've got a membrane responsible, or if you've got a bunch of enzymes and other things responsible for pumping protons, and pumping protons is the main function of the mitochondrion, then you're probably putting those on the super folded up membrane that's been all folded to maximize membrane surface area so you can fit more of the enzymes responsible for cellular respiration into the same small package. Cool? Everything tracking so far? Any questions? All right. There's 3 stages of cellular respiration. Glycolysis, which you've seen the less simplified version of glycolysis. For us, this is glycolysis. There's a glucose molecule, and it breaks down into 2 molecules of pyruvate. And you get a tiny bit of ATP. That's all you need to worry about, though you've seen that glycolysis is actually a very complicated process. All right? Cool? So glycolysis. You take a glucose molecule, which is a 6-carbon molecule, and you break it down into 2 molecules of pyruvate. You went from 1 big thing to 2 smaller things. It's a catabolic reaction. It's exergonic. And you are going to get a tiny bit of ATP, okay? A tiny bit of ATP via substrate level phosphorylation. Step 2, all right, is step 1 can occur actually outside of the mitochondria. Think about it like this. That glucose molecule is really big. It's going to be difficult to ship it across the outer membrane of the mitochondrion. That's not actually like 100% accurate, but I'll tell you, it'll help you memorize it. Like which step happens outside of the mitochondria. It's the step that takes the really big molecule and breaks it down into 2 smaller molecules. Okay. So glycolysis, you break glucose down into pyruvate. A single 6-carbon sugar becomes 2 3-carbon molecules. All right? Oxidation of pyruvate / the citric acid cycle. Usually just referred to as the citric acid cycle or the Krebs cycle, okay? Yes, this is where you take the pyruvate molecule and you break it down to carbon dioxide. So after the Krebs cycle, or the citric acid cycle, you've completely broken down pyruvate. And therefore, you've completely broken down the glucose molecule, because remember, the glucose molecule gave you that pyruvate. So you go from a 6-carbon sugar to 2 3-carbon molecules, which then break down into 1-carbon carbon dioxide molecules. So you've gone another set of catabolic reactions, and more energy can be harvested. Okay? Once again, in step 2, you get a tiny bit of ATP via substrate level phosphorylation. In step 3, this is where you get the big payoff of ATP, all right? So the big payoff here comes from oxidative phosphorylation, which is an umbrella term including both the electron transport chain, which is when the electron goes boop, 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 and pumps protons, and then chemiosmosis, which is when those protons go through ATP synthase and they can go vroom. I'm making more noises today because I know that all of our energy levels are a little low. I hope that it's working for you. All right, here's cellular respiration. All right. If you memorized, if you knew what this figure meant and memorized it all, you'd do pretty well on the respiration questions on your next exam. I'm going to give you lots more detail, but this is a good figure because it summarizes everything, you know? Like, if it's me and I was doing this for a course, I would take this figure, I would put it onto the center of a piece of paper and print it out, and then annotate this figure with all of the details about, like, oh, I'd make a circle around this and say, this is where most of the energy is by the time step 2 ends, and things that I emphasize as important to you. Anyway, that's just like how studying works for me. Here, look at the figure. Those 3 steps that I showed you before. Check it out. Step 1, glycolysis. Pyruvate goes in. Step 2 is pyruvate oxidation and the citric acid or Krebs cycle. Step 2 here is going to finish breaking down into carbon dioxide, okay, and get you a little bit of ATP. If we're going with the number of 32 ATP per molecule of glucose, you get 2 here, you get 2 here, you're going to get 28 over here. So when you're done breaking down the glucose, all of the energy, or the vast majority of the energy, is actually stored in these high energy electron carriers. All right? So these molecules, NADH and FADH2. FADH2 is basically just worse NADH. It's like lower energy, but it does the same thing. These molecules here, they're going to take an electron and still keep it at a very high energy level, and then they're going to go over and reduce something, all right? And then they'll become oxidized. So when it's NADH, it has the electron, but then when it loses that electron, it loses an H, and it ends up with a net positive charge, because it just lost 1 unit of negative charge. So then it's NAD+. Don't worry, I got slides about it. Hey, there's a video linked in your PowerPoint, and you can watch it. It's like 4 minutes long, but I'm not going to play it in lecture, all right? It's a good video, but there's 1,000 videos just like it that explain cellular respiration, and you can absolutely watch them when you feel like it. Cool, pick the one you like. If you find a really good one, share it with your friends. When you go back home to visit, show it to everybody. Go out to the bars, put it up on your phone, you know, hey, look. See, look, the pyruvate's smaller and it can go across the membrane. I don't know. Maybe you'll find love. So here's the thing, right? We're going to look at each of these steps in a little more detail. Ready? Glycolysis. Glucose. Each one of these gray circles indicates a carbon. 6-carbon molecule. Break it down. You get 2 3-carbon molecules. All right? You get a little bit of ATP. You also get some NADH. Both of these molecules represent energy. All right? ATP is straight up like pure, it's very accessible energy. It's like, oh, I can use this energy to drive all these different processes. Pumping stuff, moving motor proteins, a third thing, all that, right? NADH is more like reducing power. this NADH molecule has the power to reduce something. And when you reduce something, you can, bless you, build something bigger, all right, by attaching electrons. That was not a sneeze. That was a bad response. I'm very embarrassed. Yeah. I hear a noise, I just say bless you now. Yeah. NAD plus, NADH. Thank you. That's very good. It's got NADH. This has reducing power. So it has the ability to take that a high energy electron and put it onto something else. Okay? Cool? Awesome. Glycolysis, substrate level phosphorylation. It's very fascinating. If I was further along in the lecture, we might talk about substrate level phosphorylation more, and we might talk more about glycolysis. So here's another glycolysis pathway figure. Who asked if I had the actual one? That's you? Yeah. Everybody just pointed at you. Very good. No, it was good. I'm pretty sure this is the full pathway right here. So it's not, it's not as bad as some pathways, but to just go from glucose to pyruvate, which is basically you just broke it in half, it's pretty intense. Yeah, exactly. This is not all of cellular respiration, right? So there's these energy investment steps, and then these energy return steps in this pathway. It's a very annoying thing where it actually initially costs you 2 ATP molecules to carry out glycolysis, but you get 4 ATP molecules. So there's a net gain of 2 via substrate level phosphorylation. I know, it's absolutely fascinating. Absolutely. Here's a slide that I would like to end on, okay? NADH and NAD+. So here's NADH. Oh, I'm sorry. No. This is NAD+ right here, okay? And what it can do is it can take on an electron in this region, okay? When it does so, it can gain a hydrogen atom, all right? It's got energy there. It can go and put that electron on something else. So our 2 big important molecules here are going to be NADH and ATP. All right, solid. And we're going to come back to this on Monday, and then we're going to talk about all of the steps of cellular respiration. It's going to be a ton of fun. You're going to love it. Yeah. Of course, thank you. Have a great weekend. You too. Yeah, it's Friday. Have a good one, everybody. I'll see you all soon.